Now, I mentioned Dr. Mary Schweitzer, and this is a little complicated, but I really want to show her work because she has pinpointed actual organelles inside the cell. Okay, you lost me. What are you talking about? Do you have organs in your body? Yeah. You have liver, kidneys, right? You have a stomach, intestinal tract. These are all organs, right? Did you know your cells have organs? They're called organelles because they're tiny. And what Dr. Schweitzer did is she used that immunity and fluorescence to target DNA. Here she has 3 samples, okay, ostrich, Brachylophosaurus canadensis, and T-rex. So there are 3 osteocytes here. Do you see them? These are cells that came out of the bones. So here's a T-rex bone cell. Here's a B. canadensis bone cell. And here's an ostrich. This came from a living organism. So she's comparing the dinosaur cells to their closest relative, birds, right? Some scientists believe birds are dinosaurs, or they descended from them. And she stained them with anti-DNA. So she used that immunity Lego complex to go in and look for and attach to DNA. Look at the concentration of DNA in what is probably a nucleolus in this cell. 67 million year old cell showing DNA. This is her work. This is all published in the open literature. B. canadensis, same thing. Look at the return on the living cell, or close to living cell. Much bigger, right? A lot of DNA. Why? This was just alive. These guys, 4,600 years old, 5,000 years old, right? And they still have DNA in there. How does this happen? This is impossible. I'm showing you impossible pictures today, according to what I was taught as a scientist. Here's another one. This is propydium iodide. This is another DNA stain. And once again, these are T-rex. These are B. canadensis. This is ostrich. So there's the nucleolus. Here's the nucleolus with DNA. Look at it in the ostrich cell. Much bigger response. Why? This was just living. See? Same thing here. She used DAPI. So she used 3 different DNA stains, and every single one of them returned a signal. Okay? This rocked the scientific world. And it actually got some people in trouble, not just myself. Because now people are realizing, holy cow, this is real. These original proteins are real. They're still there. So what did they do? They said, okay, you have proven that these are dinosaur soft tissues. Now find a mechanism. How is it still here? Show us a mechanism. Can't be a God mechanism, right? That's the M word, miracle. Can't go there. Find us a scientific reason for why these tissues are still here. And so they've fallen upon iron. They say the iron from the blood, okay, preserve these tissues. This is why you need to go back and watch the earlier presentation, because I spent 15, 20 minutes showing you that all these bones are clotted. Every single one of these bones have clots, blood clots in them. You got to go back and watch that presentation because it shows, number one, that the iron was clotted in the bone and not available to preserve tissue. And number two, it's a condition of drowning. Blood clotting is a condition of drowning victims. This is well established in the medical literature. It's called disseminated intravascular coagulation. You go through a traumatic event where you're being asphyxiated in water and your blood will clot. Interestingly, that's a mechanism that COVID uses. Did you know that? COVID is causing systemic clotting. I find that really compelling. Nevertheless, those blood clots that we found show that all of these dinosaurs, and every single one that we've tested for blood clots, have shown blood clots. They all drown in water, just like the scripture says. All right, so osteocyte cells. They're not contamination. I mean, seriously? These cells are going to crawl across the dirt and crawl into a bone that they weren't living in and set up shop there and so I can find them just to say, oh, it's contamination from another organism. Come on. Seriously? They have minimal birefringence. They don't glow in polarized light, meaning they're not rock. They haven't hardened into rock. Okay? They're not biofilms. The cells autofluoresce and take up vital stain. They take up nucleic acid stains, and they take up immunohistochemistry stains, as I showed you. But they don't behave like normal tissues do. Biofilms do not permineralize. They do not turn into rock. And we find some osteocytes that are permineralized. So this is not biofilm. The reason I talk about biofilm is because it's still a prevalent theory. There was a brand new paper published at the end of last year by a group at University of Washington in Seattle. This is their second paper trying to prove to the scientific community, nothing to see here. These are just biofilms. But they didn't really do the kind of work that we have been doing to show that they're not biofilms. So it's an accepted theory, and that's why I keep talking about the differences. Here's the paper I was talking about. Thomas Kay, he's at the University of Washington. This is his second paper on biofilms. And I only talk about this because when you go out and you start sharing dinosaur soft tissue with people, you're going to hear biofilms. So I want you to be prepared for that. So they claim these tissues are only biofilms. They did identify several different bacteria sequences. So they have identified the different bacteria and microorganisms that are in these bones. And one of the comments they made in their paper was, there is more life inside this dinosaur bone than in the soil around it. Now, why is that? Why would there be more microbes in the bone than in the soil? Use your common sense. There's food in them there hills. And that's what they're going after. That's why these bones are so full of bacterial communities. So they're actually arguing against their own theory when they say this. There wouldn't be bacteria in here if these are 67 million years old because there wouldn't be any food. You follow me? Okay. They mostly used elemental analysis. What do I mean by that? They were just testing for the elements, you know, the magnesium, the calcium, all of the different elements. So they didn't do a lot of microscopy there. But again, they proved there's more nutrients in the bone than in the soil around it. And their only microscopy proves my point. They did a test, much like Dr. Schweitzer did, much like I did, where they looked at a blood vessel in regular light, and they looked at a piece of bone in regular light, and then they stained them with propydium iodide for what? RNA, remember? So they're saying that these have to be biofilms because they're RNA in them. But I did the same tests with the tissue that we pulled out of the bone. So they are not biofilms. This is a picture that simply shows how the cells will fluoresce, but the biofilm really doesn't. So only the microorganisms stain in a biofilm. And they're simply showing that there's a lot of RNA in their sample. That's all they're showing. And it's funny because they don't talk about that, but that's the implication of what they're showing.